For reversed phase, it's a little bit different because we now have to take into consideration pH of the mobile phase. So for acidic conditions, we can still use the additives that we highlighted before, TFA, formic acid, acetic acid. You just need to make sure that the mobile phase is pH 2 or higher. Less than pH of 2 will start to potentially decrease the column life. For a neutral molecule, just running it with water. And for basic molecules, we need to move away from using the organic bases like TEA and DEA and move into something like a buffered ammonium bicarbonate or ammonium formate solution. Bicarbonate, formate, acetate, all are safe for the column at pH 9 or less. If you use a phosphate buffer, which is safe for the column, you do need to make sure that it's actually pH 8 though. Phosphate's a little bit harder on the column, and so we want to make sure that we, again, extend the life of the column. As far as concentration goes, 20 millimolar is pretty common, but you can go up to 100 millimolar. You just have to make sure that the buffer is soluble in the organic modifier that you're going to be using. So you don't want to go too high. Buffer could precipitate out on the column and cause some damage. Now, I mentioned we want to move away from using those organic bases in reversed phase because the pH of the mobile phase is now going to be far too high for the silica. So under normal phase conditions here, we're really not concerned about pH. It's an effective pH that's not really of concern. Under reverse phase conditions in aqueous mobile phase, 0.1% TEA in the mobile phase will give you a pH of 12. And pH of 12 is strong enough to begin to dissolve the silica gel base material. So even though you might have a nice separation initially, you will begin to destroy the chiral stationary phase. Method development is very similar to normal phase in that the percentage of the polar component, in this case we're looking at the organic, either acetonitrile or methanol, is going to push things off faster, so higher percentage is going to push things off faster. 60-40 water acetonitrile with TSO here gives you a method of almost an hour, but dropping the percentage of water to 50%, going 50-50, now you're beginning to get things off the column faster. Here, 40-60, 30-70, 20-80, and all the way down to 10-90, co-eluting everything all together. So similar principles though as with normal phase. What makes for a challenging investigation? This is a question that I get asked quite often. We do find hits on most of the molecules that we screen. Usually it's somewhere in the 90-95% range. However, these are some of the things that make it a little challenging. Large molecules can be challenging in that they oftentimes have the stereocenters buried within a lot of other functional groups. And so it's hard for the molecule to actually get into those chiral grooves that I showed before. Multiple chiral centers are challenging oftentimes because there's just too many peaks. You're talking about 2 chiral centers, the potential for 4. You're starting to look at 8, 16 enantiomers or diastereomeric pairs. That's a lot to try to discriminate on a chiral stationary phase. So 2 peaks is usually pretty easy. The chiral center is too far away from good points of interaction. So if you've got a phenyl group, which is a pretty good hook, as we would call it for a chiral stationary phase, but the chiral center is 5-6 bond lengths away, it's not going to be advantageous for finding a good separation. That chiral center is simply too far away from that good point of interaction. Or you have too many or not enough interactions, or too many different interactions. And so either it makes it really challenging to find what additive is best, or you have too many interactions, things are just coming off the column far too slowly, things are broadened extremely significantly. So this is kind of an all-inclusive example of those. This one is complicated by far too many interactions. This has stereocenters that are buried within the largeness of the molecule, to make a word up there, not easily distinguished by the chiral stationary phase, and it's unclear what proper additive should be. I won't show you the molecule yet, but I'll show you the screening. We did this under reverse phase conditions on a gradient, as I mentioned before. This is on chiral cell OZ3R. You actually have almost a baseline resolution of some of those peaks. Or I'll show you the molecule now. It's a 36 mer polypeptide. It's 4 isomers. There's 2 amino acid variants. They were buried within this chain. One, I believe, was towards the end. It was either 34 or 35, and the other one was in the middle here at 15. If we zoom in on the separation, we do have close to baseline resolution of these last 2 peaks. I would venture a guess to say that those 2 peaks are probably the variants at the end of the chain. So you were seeing some discrimination because there was access to the stationary phase of that particular stereocenter. That was a variation. But then you have coelution here of 2 other peaks, which are probably the variants that are in the 15 position, simply too buried within this molecule to actually reach within the chiral groove to see some separation. The addition of acid is usually helpful for polypeptides. Like I mentioned before with amphoteric molecules, acid is usually helpful, but in this case, there's so much else going on. You've got basic functional groups, you've got acidic functional groups, that TFA by itself is simply not sufficient enough to give you good separation, or in this case, you do see some peak tailing. In the case of polypeptides, using a cheotropic salt like potassium hexafluorophosphate oftentimes is helpful. It helps stabilize the linear conformation. It gives you access to those buried stereocenters, but in this case, it was not helpful just because of how large this molecule was. And in some cases like this, you might even need to look at coupling columns or developing some experimental design to help test other method parameters. In this case, we were not able to optimize the separation any more than what we achieved here. Either the peak tailing was too significant as we tried to retain the compounds, or we just lost all chiral separation whatsoever. So these are challenging molecules, no doubt. A couple more resources for you in addition to those that are up on our website. We've got a couple papers from our colleagues at Chiral Technologies Europe. Here, this is for normal phase immobilized polysaccharide screening. We have one for reversed phase strategies as well, also from our colleagues at Chiral Technologies Europe.